The overall goal of this procedure is to study and compare the virulence of Mycobacterium abscessus strains and to describe the interactions between these bacteria and the host innate immune system in vivo using transparent zebrafish embryos. This is accomplished by first preparing homogeneous bacterial inocula. Next, the embryos are intravenously injected with the bacterial suspensions at 30 hours post-fertilization. Additionally, to study more precisely the role of macrophages in the infection control, a lipoclotronate-based injection procedure is used to generate macrophage-depleted embryos. Finally, the infected embryos are prepared and imaged using fluorescence and confocal microscopy to monitor the progression of the infection. Ultimately, the results which can be obtained show the specific interactions between the fluorescent phagocytic cells and M. abscessus, either as individual bacilli or as serpentine cords inside a transparent embryo.
The main advantage of using zebrafish over other animal models like mice is that it allows to specifically investigate the infection process in real time and to study and image the specific interactions between Mycobacterium abscessus and macrophages or neutrophils.
This step is a critical issue for the subsequent injection procedure in zebrafish due to high propensity of rough Mycobacterium abscessus to form large aggregates and to produce cords. A specific treatment is therefore necessary to prepare homogeneous and quantitatively controlled inocula prior to microinjection.